Hi, this is Stacy Tannenbaum. I'm a Senior Director in Pharmacometrics at Astellas Pharma, and welcome back to Part 2 of Module 2, a review of the various model types commonly used for MIDD. Part 1 of this presentation focused on PK principles and modeling. So we really focused on the PK components because that's foundational to understanding the PK/PD. If we don't understand the disposition of the drug and the exposure to the drug, then everything downstream is going to be difficult for us to quantitate and to optimize. So what you have to look forward to in the next session is an expansion beyond PK. First thing we're going to talk about is no PK and the disease progression and systems biology modeling that I spoke about earlier, which actually are understanding the disease without any intervention. Then we'll look at how the drug impacts disease progression, how the drug impacts pharmacodynamics on a rather macro level, and how the drug impacts pharmacodynamics on a micro level by talking about quantitative systems pharmacology. Disease progression can be extremely complicated. Patients don't just uniformly get worse or uniformly get better from a disease or from an illness. Sometimes they can impact their own disease progression in both a positive or a negative way. Patients who have, for example, high blood pressure, cardiovascular disease, diabetes, might be able to impact their disease in a positive way by changing their diet, starting to exercise, stopping smoking, etc. There's also other medications that they might take that would change their disease progression separate from the drug that we are studying. And of course, patients can get worse over time without any intervention or potentially take some interventions that make their disease progress faster. So diseases can change without intervention or with non-pharmaceutical intervention, and it's up to us to make sure that we understand that so that we can subtract off these external factors from the impact of our drug that we're studying. In addition, we have to consider things like the placebo effect. So this is a non-pharmaceutical intervention, but that can have a very high impact on disease progression in particular diseases. So again, we have to be able to subtract that off from our understanding of disease progression in the presence of our drug in order to understand specifically and in order to quantitate how much our drug is having an impact on the disease. Talk about disease progression, we often think about the endpoint, which is generally defined as how the patient feels, functions, or survives. Endpoints are generally something we can easily observe or measure. Does the patient feel better? Is their blood pressure reducing? Is their tumor shrinking? Can they remember more words in a memory test or have more steps in a 6-minute walk test? But diseases are often complex and multifactorial. They can impact other seemingly unrelated systems and pathways in the body. In cases where we need to have a granular understanding of up or downstream effects or feedback systems during diseases, or when important biomarkers or other factors can't be directly measured, we might need systems models like these to understand the organism as well as the disease at a more micro level. I'm not going to speak much more on this at the particular moment. I'm going to come back to this concept when I cover quantitative systems pharmacology. That marries these disease models with pharmaceutical intervention. For now, I'd like to focus on the more simple disease progression models and how treatment can impact these trajectories. So let's start with a really simple example where you have a linear progression of your disease. That looks like this line here. So in this particular case, we have two components. One is the baseline disease state. You want to think of this as, let's say, the tumor size or the blood pressure measurement at the time that the patient seeks intervention. Obviously, if you went all the way back in time, you would not have a blood pressure of 0, but you would have a tumor size of 0. So really what we're looking at is the time that the patient goes to seek intervention. There's also a natural history, and we know that not all diseases will progress in a linear fashion or get worse and worse and worse forever, but let's, for the purpose of this example, take this as a period in time where the disease is progressing linearly. Now, if you add active treatment, and this is the bar that shows the time of active treatment, and again, the time on the x-axis and the disease severity on the y-axis are just there for simple example. Keep in mind, there's also the placebo response included in this We haven't taken into account the placebo response. There are different ways to subtract that off by looking at, for example, a placebo arm in a trial versus a non-placebo arm in a trial or a treated arm. But for the purpose of today, we're going to call active treatment be the combination of the active treatment and the placebo response. There's a couple of different ways that active treatment can change the progression of disease. And one is probably the simplest, and that's symptomatic relief. So that is when the patient is on the treatment, they feel better. Their blood pressure lowers, their tumor shrinks, but it doesn't necessarily change the course of disease progression. And when the patient would go off the treatment, they would essentially return to their normal disease progression that they had before. There's also the situation where treatment actually modifies how the disease progresses. So in this particular case, consider a tumor that is growing in a linear fashion, and what you've done is you've reduced the slope, you've slowed down tumor progression. Obviously, if the tumor shrinks, you would have a negative slope, but in this particular case, we're just looking at a slower progression of disease. Again, once you go off the treatment, if this is a reversible effect, and I should have mentioned that in the previous slide as well, these assume reversible effects, your disease progression would go back to the rate that it was previously. And in this particular case, you see a combination of the two types of disease modifications that you saw before. You see a symptomatic effect where the patient starts to feel better, the disease severity goes down, and you also see a reduction in the slope. So the disease is progressing in a slower fashion. Of course, there's many different other combinations that you can imagine, but these are the primary ways that a drug can impact the progression of a disease. So what I've been describing thus far is kind of the most simplistic way of thinking about how an intervention impacts a response. In the previous examples, we really talked about intervention as a yes or a no, and then what happens downstream is a function of whether or not there has been an intervention. Pharmaceutical interventions do indeed lead to responses, ideally, if our drug works, of course. But it's a very rare situation when the response is just a yes or a no. Generally, the responses change with dose, and that's why we need to understand the PK-PD relationship. If we change our dose, our formulation, our dose frequency, you give the dose with food, you give the dose with other medications, you need to understand how that's going to impact the downstream response. So how you tie PK and PD together is exposure. And we're going to talk a little bit more about what that means in the next slide. So there are a number of exposure metrics that we can consider for looking at our exposure-response relationship. And one, of course, is dose. But generally, we look at something slightly more downstream, such as the area under the curve, and that can be the AUC from 0 to infinity, or during a certain time frame, like one dosing interval. We can look at Cmax, the maximum concentration. We can look at Ctrough, the minimum concentration. We can look at an average concentration. We can even look at concentration at a specific time, and generally that time is at the same time that you would be measuring a PD response. It's important to note that different endpoints may correlate with different exposure metrics. Rarely do we have a single exposure metric that can describe every response. And we have a number of responses. We don't just measure the efficacy response, the blood pressure, the tumor size. We measure biomarkers. We measure safety endpoints. And so we need to really explore our exposure response for all of these different endpoints that we measure. It is not 100% true, but a general rule is that the efficacy is generally correlated with the AUC, whereas the safety is generally correlated with the maximum concentration. But again, this is a general rule, but certainly not the same for every compound. It's important for us to know exposure targets and caps when we're trying to optimize our dose. You want to stay, of course, below your exposure cap where you've seen some AEs in our preclinical studies. And you also want to stay within a therapeutic range or above a certain target range, or sometimes below it, depending on what is your endpoint. So for example, the minimum inhibitory concentration is something that's used very often in ID, infectious disease, where you want to be above that minimum inhibitory concentration for a certain amount of time. And exposure matching is very important when you're doing things like driving scaling. So we often will calculate a pediatric equivalent dose, i.e. we calculate the dose that is required to get the same exposure in a pediatric patient as what we saw in adults. And that's often our first step on optimizing pediatric dosing. Now, exposure response can be challenged by a number of different components, and one is the fact that, and repeating what I said before, different endpoints may correlate with different exposure metrics, and it's up to us to really figure out what is the optimal exposure metric for us to be able to then make predictions downstream on what would happen if we change the dose to various different responses. So just from a time and resource perspective, it can be very complex, particularly in studies where you have a lot of different endpoints. Downstream effects also may not correlate with temporal exposure, i.e. the exposure at a certain time may not beget the response at that exact same time. Sometimes you have a delay in your response, and it can be quite a long delay sometimes. And so you would need to incorporate some kind of models, transit model or lag times, in order to account for delays. And I'll talk about those a little bit when I start talking about the individual models. And of course, it's not always the concentration in the plasma, which is what we generally measure, that drives the response. It's often the concentration in the tissues or the joints, at the site of action. And these may be difficult, invasive, or impossible to measure. But of course, you would be not surprised to see me say that modeling can be utilized to be able to estimate things that we can't necessarily measure. So if it's something, for example, the joint concentration, if we understand enough about the PK and how the drug partitions between the plasma and the joint, we could estimate what that looks like. Also, we can use modeling to adjust for that temporal correlation, and I mentioned that already, or if we need to calculate the concentration at the time of the event. Sometimes an event happens and we didn't measure a concentration at that time, but if we have good models for the individuals, we should be able to interpolate the concentration for that individual at the time that they had an event, which will give us a better idea on whether or not these two things are correlated. So speaking of models, let's start talking about PK/PD or exposure response models. What you see on the left side is a concentration time profile, and what you see on the right side is an effect versus time profile. So in order to figure out our exposure response relationship, we first have to plot the data. And we do that by taking the concentrations and the effects at the same time and plotting them versus each other. And very frequently, you will see a shape that looks like this. This is the very typical Emax model. I'll be talking more about the Emax model later. I should note, though, this is not the only PK/PD profile shape that you will see. There are a number of different profile shapes. This is the most common, but really it depends on whether or not you are able to characterize the entire exposure response curve. If you have a very, very wide dose range, and particularly in preclinical studies you might be able to characterize this whole thing. In a lot of other cases, if you're not seeing this full curve, it's not because it doesn't exist, but very likely it's because you haven't been able to collect the doses that are either high enough or low enough to be able to characterize this curve shape. So since the Emax model is the most common of the PK/PD models and very likely underlying most of the PK/PD that we see, whether or not you can characterize the entire profile, I'd like to spend a little bit of time on that. And I think the Emax model is useful because it characterizes what we expect to see from PD, a continuous monotonic relationship reaching a maximum drug effect at sufficiently high exposures or doses. So let me orient you a little bit to the Emax model. If you watched part 1 and you remember when I talked about the Michaelis-Menten model, and I mentioned that you'll see a very similar functional form for Emax, well, that's what you see here. This will look very similar to the Michaelis-Menten model that I showed you before. So let's start with the parameters of an Emax model. And we start with E0 and Emax. E0 is your minimum effect or your baseline. Depending on what is plotted on the y-axis, you could have raw values, in which case, let's say for blood pressure, you're not going to have a baseline of 0, but you're going to start with some kind of minimum effect. Keep in mind, This plot can also be flipped upside down. In the case of blood pressure, as I mentioned, you generally want to reduce it rather than increase it, in which case you would want to see those values go down with increasing concentration. But for the moment, let's consider our Emax to be in the positive direction. We also have a maximal effect. At some point, you will not get more effect no matter how much extra drug that you put into the system. Because what's going to happen is, let's say if the drug needs to bind to a receptor, you've already maxed out all of the receptors. You've already maxed out target engagement. Your body just cannot increase your effect any further. If you continue to put drug into the system, you may see some off-target effects or AEs, but you're not going to get more than 100% of your possible effect. So the Emax is often considered to be 100%. Sometimes, of course, you don't reach that 100% value. EC50 is also considered the potency. So this is the exposure needed to obtain 50% of the maximal effect. In this case, I took the difference between Emax and E0 to be 50% of that. This is an important parameter because it helps us to understand how potent our drug is. If you have a lower EC50, you need a lower exposure to obtain 50% of the maximum effect, i.e., you have a higher potency. And last but not least, there's this gamma parameter, and that represents the steepness of the curve. What you can see on the right side is often called the sigmoid Emax model, so it has more of an S shape than the shape that you see in the original plot. In the particular case on the right, where gamma is greater than 1, the same change in concentration will give you a much higher change in effect than if you looked at the same plot on the left-hand side. So that's the Emax model, but really the selection of your model is going to be based upon a number of different things. Emax is very common, but we often will choose our PK/PD model based on the mechanism of action, the temporal relationship between PK and PD, whether the concentration at a certain time begets a response at exactly the same time or whether that happens downstream, and the type of PD data. I just want to go over some of the things that we consider when we're choosing our PD models. What you see in red is representative of that Emax model that I showed you before. So we assume that the type of endpoint is continuous, that there is a direct effect, that the effect is reversible, and that there are no delays, tolerance, or sensitivity. But like I said, while this is very common, there are always exceptions to the rule. So if we go back to the type of endpoint, continuous is quite common, but sometimes we look at things like time to event models, count models, so how many events you had within a certain period of time, for example, binary models, do you have the event or do you not, ordered categorical models such as pain or disease severity. Sometimes we have indirect models. So if you bind to the receptor, that often causes a direct response. But sometimes we can look at things like impacting the buildup or loss of the response mediator, and that's not impacting the effect itself, but something upstream or downstream to it. So those are indirect effects. There are irreversible effects, especially now with many of the gene therapies, we're actually changing the fundamental components of the body. These are not reversible effects like a drug binding to a receptor and then eventually coming off the receptor. I mentioned delays. That has something to do with the indirect effect, but you can also have things like distribution to the site of action, signal transduction, enzyme inhibition or induction that then cause a downstream effect. It's a little bit related to the indirect effect, but we can also have just timely delays. And I will talk briefly about tolerance and sensitivity development because those can impact whether the same concentration has the same response at a different time. So let's start with the concept of the hysteresis loop. What you see here is an effect concentration plot, but it looks a little bit different than what we saw before. What you see here is a loop, and that loop is time. So I've just noted 2 times on here, time 1 and time 2, where time 1 is earlier than time 2. And what you can see is that at the same concentration at time 1 and time 2, you see a higher effect at time 2. And this is often due to delays. So these are situations where you have drug distributing to the site of action. So even though the concentration in your plasma is the same, the concentration at the site of action is lower at an earlier time because the drug hasn't fully distributed to that area. It can also represent a downstream response, so that's just a time delay, or the formation of an active metabolite, which is also contributing to a time delay. This can also be a function of sensitization, which really doesn't have anything to do with time, but has to do with the fact that the patient becomes more sensitive to the drug. So later, when they see the same concentration, they have a higher effect. We often model this phenomenon by using the schematic that you saw several times in part 1 by putting in what we call a hypothetical effect compartment. That might be realistic. It might be that, for example, the joints or the tissues, the concentration in those tissues is what drives the response. But generally, we use this as a way to collapse that delay. If we plotted the pharmacodynamic effect versus the drug concentration in the effect compartment instead of in the plasma, what you do is you collapse that loop into a more traditional Emax model, for example, and then you can do your modeling from there. In addition to counterclockwise hystereses, we occasionally will see a clockwise hysteresis, where at a later time you may see a lower response than you see at a earlier time. And that often is the patient becoming tolerant to the drug. Obviously, for things like sensitization and tolerance, if you can understand the mechanism behind those, you can certainly work those into your models. But we do have ways of accounting for them in an empirical way with things like the hypothetical effect compartment and some other model-based ways of explaining these phenomena. The hypothetical effect site model that I showed you previously allows for distributional delay in effect, but it assumes that there is a direct effect between the effect site concentration and the response. So what indirect response models assume is that the site of action either precedes or follows the measured response variable. We assume that the drug effects can inhibit or stimulate the factors controlling either the input or the dissipation of drug response, but not the response itself. So in an indirect response model, we can have inhibition of the production of response, which is going to make the response go down, or we can have stimulation of the production of response, which is going to make it go up. And I'll show you some schematics in a moment. And similarly, we can do the same thing for our loss of response. We can have inhibition or stimulation. So this is what these plots look like. One thing that you're going to notice is that we have plots that look kind of similar in structure. So if we have a production of the response being inhibited, the response will go down. If we have a loss of response being stimulated, the response also goes down. So it's very important when we're going to be using indirect response models that we're using them responsibly. You should have an idea of the mechanism of action of the drug, because if you use the wrong model, even if it might fit the shape, this may give us wrong predictions as we go downstream. It is actually a useful tool, though, for comparing competing models and their predictions. We could bring those forward as part of a hypothesis generation if we're not 100% sure of how this particular drug works. But generally, you don't want to use the indirect response models unless you have a reasonably good feel for what is the mechanism of that particular drug. So we've dipped our toe into the water of semi-mechanistic models by looking at the indirect response models. Another one that you may see fairly commonly is the TMDD, or target-mediated drug disposition model. If you see concentration time profiles that look like this, where at the lower doses you see a much faster decline in PK than you see at the higher doses, as well as this strange nonlinear shape, it's very likely due to the fact that you have target-mediated drug disposition. What this means is that when the drug binds to its target, the actual active binding impacts the PK of the drug. It may clear the drug faster or have some kind of impact on the drug's disposition. This is often, if we don't have measurements of both the drug as well as the target binding site as well as the complex, we often will represent this with an empirical model, which is just looking at the antibody and then assuming that you have a parallel linear and nonlinear or Michaelis-Menten clearance. But if you've been able to measure some of the other components, if you have, for example, the binding target as well as the complex, you can actually understand this in a much more mechanistic way, where as the antibody binds to its target, this causes a complex, and the complex has a different clearance or the same clearance as the antibody, and that can actually affect the PK of the drug. This is a situation where your choice of model is going to be dependent on the data that you've been able to collect. If you're not able to collect all of the components that you need for the semi-mechanistic model, the empirical model that you see here is a good substitute. And essentially that Michaelis-Menten clearance is accounting for the mechanistic components that you would normally be able to characterize if you had collected the appropriate data. But I think this is a nice example for you to see that sometimes we can only use an empirical model, but if we have more data, we can always choose a mechanistic model, assuming that we have a good understanding of the mechanism of the drug. It's definitely a more informative approach. Another semi-mechanistic model that is often used in the oncology space is the Freeberg model for myelosuppression. It's named for its author, Lena Freeberg. This model establishes a relationship between drug concentrations and myelosuppression, which is dose limiting for many anti-cancer drugs. So the model looks rather complicated, but it's actually fairly simple. It's got three components. One is a proliferating compartment that is sensitive to drugs, and we'll talk about that part in a moment. There's three transit compartments that represent maturation as a function of time, and then there's a compartment of circulating blood cells. And those circulating blood cells feed back to the proliferating compartment. So an overshoot compared with the baseline value will reduce the production of stem and progenitor cells. So the parameterization is actually fairly parsimonious. It's really got the initial circulating concentration, the mean transit time, and this feedback parameter. So it's well suited for sparse data. The drug effect can be implemented on the proliferation compartment. It can be implemented as a linear function or an Emax model or any of the other models that we talked about previously. What this does is add a clearance mechanism to the progenitor cells. By clearing these out, they don't reach maturation and get into the circulation, and this is the cause for neutropenia so often seen with oncology drugs. So it's a rather simple model, but it is representative of the mechanism. Now, these transit compartments don't represent actual reality in terms of all of the steps in the life cycle of a maturing cell, but they're close enough to be able to represent the delay and the timing between the proliferation compartment and the circulating blood cells. So this is why we call it a semi-mechanistic model. Later, when we talk about QSP, you'll see the difference where we talk into full mechanistic models. But we're getting there soon, so stick with me. I just want to touch on a couple of additional models before we wrap up PK/PD. And one is for binary responses, yes or no, live or die, respond or not respond. And these are generally modeled by logistic models. We model them as a probability of the event occurring. So if your data looks like this, you have a probability of 0, meaning a non-responder, or a 1, meaning a responder, you can certainly put a linear regression through it, but there's a couple of problems with the linear regression. First of all, you don't have a graded response. And also, your values can go below 0 or above 1, which for a probability model is not appropriate. What a logistic regression does, or a logistic model, it allows you to constrain the values between 0 and 1, and you see more of a graded response. So you can see that it's more appropriate that at the low exposures you have more non-responders, at the high exposures you have more responders. Generally, this is pretty clean data. You're going to see a lot more overlap in data like this. What's useful about a logistic regression model is if you're trying to, let's say, get a certain probability of response, let's just say 75% response, you can pick out the exposure that is required in order to get there, and that helps us with optimization of our dosing. And last but not least, there are the very complex time to event models. We have a couple of examples of those here where, let's say, for each individual, you will record the time at which they have an event. And then on the right-hand side, you can collapse this data into what's called a Kaplan-Meier curve. So starting with the number of patients that you have or the percentage of patients that are surviving, as patients have their event, they start to drop off. And you can do a comparison between two drugs. I'm not going to go into the details of how to model this, but we have models for time to event. We also have models for count data. We have models for graded categorical, such as different levels of severity. I think the point that I want you to take home from showing these is not necessarily that you need to become an expert in all of these models, but that PD is complicated. PD often has more variability than PK. There's different kinds of endpoints. PK is sort of one thing. It's the drug concentration either in the plasma or in the tissues, whereas PD can take many, many forms. It's much more heterogeneous than PK. And we have to understand for every endpoint an understanding of the mechanism of action of the drug, selection of the appropriate exposure metrics for each of the drug effects, efficacy and safety. There's also quite a lot more variability within PD. You might get everybody to the exact same exposure, but you will still have a wide variability in response. So it's up to us with modeling and simulation to be able to characterize as much of that variability and as much of that mechanistic understanding as we can in order for us to use PK/PD to optimize our dose. We've spoken about empirical PK/PD models, we've spoken about semi-mechanistic models, and now let's talk about mechanistic models, and that's under the category of quantitative systems pharmacology. QSP provides a holistic quantitative understanding not just of the drug disposition, but the relevant disease pathology and the drug-disease interactions. And you always start with this systems biology model that I've spoken to earlier in the presentation. You would model the disease on its own, understanding all of the pathways of disease. And then you add perturbations to the system by various pharmaceutical interventions and sometimes combinations. So you perturb the various parts of the system and you see what happens downstream. Now, there's no question that QSP is substantially more complicated than having an empirical or a semi-mechanistic model, but it can answer questions that traditional PK/PD cannot. We already spoke about some of the downstream effects of a perturbation at various points in the pathway, and also how that pathway relates to other organ systems and pathways in the body. We also talk about feedback loops that modify the pathway. So you can look at things that are upstream. If, for example, you increase the amount of a particular enzyme, does it go back and down-regulate its production? So all of these things, if it's mechanistic, we need to be able to incorporate that into the model. And how are other pathways, organs, and systems affected by your perturbation of this particular system? And you really need QSP and its modeling of the relationships between these pathways in order to be able to answer such questions. The beauty of QSP is that these models can be used and reused. Because they're representative of the disease state, you can put in different drugs, you can add in pathways, you can add or subtract detail as necessary, which makes QSP models, even though they take a very long time to develop, very useful for future use as well as current use. So as an example of model use and reuse, let me introduce to you the Peterson and Riggs bone model. This is quite a famous model within the pharmacometrics world, and part of the reason it's so famous is it's been under development for more than 15 years. The reason for that is because the model was originally developed to answer one question about one compound, but over time, as more pathways were added, more organs were added, more systems were added, different drugs were tried, different biomarkers were collected, the model was expanded and extended. And that's what makes QSP models so useful, because they represent not only a disease state, but you can expand on other diseases and other compounds, which really makes these models into platform models. One of the beautiful things too about this particular model is that it's open source. The folks at Metrum, where Matthew Riggs works, have made the model code available to anybody who wants to use it. And as a result, people have come in and used it for their own purposes, refined it and added to it, which really makes this more of a group effort. So QSP models, while they are not trivial to put together, they do lead to some very interesting discoveries and collaborations downstream. As I said, they are complicated. This particular model, when you look at it, it kind of makes sense in an intuitive way. You see a kidney, you see the gut, you see the extracellular and intracellular fluid, and you see the bone. You see how all of these things are connected together. But under the hood, are thousands of equations, very, very complicated mathematics. And so that's part of the reason why QSP is still finding its way within regulatory science, because it's very difficult to evaluate QSP. A lot of these parameters come from literature. You can't measure all of these, and you can't necessarily estimate all of these either. So you often are taking parameters from literature, you're taking parameters from preclinical data, and so there's quite a lot of work to do to validate these models. And there's many, many efforts going on, even within the FDA, on how to validate and utilize QSP models so that they can be used and reused without having to reevaluate them each time they're being used for a particular question. So given all of these complexities, what is the utility of using QSP within clinical development? Well, I think it was clear from showing how the Bohm model has been used is that a holistic, mechanistic, quantitative QSP model allows data obtained from one clinical scenario to inform likely outcomes of other related clinical scenarios. And actually, I would say that it's very similar to PBPK in that before you put the drug in, what you have is a system that could potentially translate from preclinical to clinical, provided that we know all of the points along the pathway that we can scale from preclinical to humans. So QSP models do facilitate translational modeling as well. Because you have a system, you can translate across diseases, you can translate across targets, you can translate across biomarkers, and I think the Bohm model represents many of these things that we're mentioning now. In addition, you can translate across drugs. Different drugs perturb the system in different ways, and this also helps us with combination products. When we put different drugs in the system, they may have different mechanisms of action that could actually impact each other. And there's really no way to understand that impact without understanding mechanistically how those drugs impact each other. You can't do that with a basic PK/PD model. And of course, we can translate across dose. Because we have a PK and PK/PD component attached to a systems biology model, you can look at what happens downstream at different biomarkers and different endpoints when you change the dose. So this is all I'm going to say about QSP models, except to tell you that you are going to be seeing more and more of these over time. Nothing will replace traditional PK modeling. Nothing will replace traditional PK/PD modeling, but in situations where you have to have a very strong mechanistic understanding of how the disease and the drug interact, you can't really replace that with empirical models. You need to be able to look at QSP modeling. You need to be able to look at systems biology. They don't always have to be as complicated as the Bohm model. It could be a very small system. It could be a single pathway. But if we have to have a mechanistic understanding of how the PK and how the drug impacts the disease, we are moving in the direction of really trying to understand these models on a very, very granular level. So be prepared. You're going to be seeing these more. Congratulations, you've made it through the modeling component of this module. So you can rest easy. We're now going to talk a little bit about simulation before we get into some examples as well as some philosophical principles on MIDD. I want to remind you that for simulation, the ultimate goal of modeling is prediction. Description is useful. And certainly, we would be remiss not to fully describe our PK and our PK/PD, but we do that so that we can understand the system better and make predictions, optimize the dose, optimize the frequency, and really understand how do we design our clinical trials. And so that's really where we are with clinical trial simulations. Clinical trial simulations, which I'll describe in a little bit more detail shortly, help us predict the probability of success with our current study design. And it helps us to determine the sensitivity of the trial outcome to controllable items. And those are things like number of subjects, number of arms, number of samples, sampling time, in other words, your study design. I actually want to pause. Normally I say number of subjects is controllable, but there are situations, for example, in very rare diseases where we think it's going to be very difficult for us to recruit for the study. So you may want to actually put that under a non-controllable item and see how sensitive your study design is going to be if you don't get as many patients as you would hope. But other non-controllable items are things like patient behavior, compliance and adherence to the scheduled dosing, the environment that the patient is in, the standard of care, and other things that you cannot control in terms of patient behavior. So those are very complicated items. Let's say, for example, you find that compliance becomes an incredibly important part of your study design. You may have to incorporate in things like MEMS caps or other ways of determining whether or not the patient has taken the medication. And of course, we can examine situations that would be ethically, financially, or physically impossible, but provide valuable information. You can have 1,000 patients in your trial. You can triple, quadruple, 100 times the dose. These are all things that you can't do in the clinic, but you certainly can do on a computer. So the first step in a virtual trial is generating virtual patients. And we can do that in a couple of different ways. We can pull whole patient demographics from things like the NHANES database or other databases that give you essentially a vector of characteristics of a patient, an age, a weight, a gender, a smoking status, and these can be real patients. We can pull them from external databases or from our own internal databases of patients that we've studied. But if you're going to generate new patients, you have to make sure that you have what's called a covariate distribution model. In a situation like this, we want to ensure that we're not pulling a 3-year-old who weighs 200 kilograms and smokes. That's obviously an unrealistic combination, at least I hope so. So we would want to build models that ensure that each time that we sample a particular covariate, such as age, it limits the body weights and the smoking status and other things that we can pull from. So these are statistical models that can be built in order to generate virtual patients that are also realistic. Then we have our input-output model, and you've seen this before. This is our PK/PD clinical endpoint model. We have to incorporate in disease progression and placebo for the reasons that I mentioned before. If we don't do that, we might end up getting a too rosy or too grim picture of what our drug effect looks like. So we have to take into account the fact that patients are getting worse with disease progression, presumably, and that they're potentially getting better when they take a placebo. And you must, of course, incorporate in variability, uncertainty, and covariate effects, which we talked about in the PK section when we talked about population PK. Lastly is our execution model. These are things like the trial protocol, the dose regimen, number of subjects, number of arms, the dosing and sampling times, making sure we incorporate in the inclusion-exclusion criteria. So as we're generating our virtual patients, it's possible those patients can be included or excluded depending upon their covariates and other baseline information. And of course, we will get a very, very rosy picture of our trial outcome if we do not incorporate things like non-compliant patients, patients who drop out, and missing data. It's really critical to try to make your clinical trial simulation as realistic to an actual trial as you can. And in an actual trial, you're going to have patients that don't behave. You're going to have patients that don't take their medication or drop out or miss visits. So you need to incorporate those in. Otherwise, you're going to get an endpoint that doesn't make sense and is not realistic for our clinical trial. When you do a clinical trial simulation, you repeat it multiple times. You don't just do it once. You can do it 100 times or 1,000 times, depending on the time that it takes and the resources that it takes to run the clinical trial simulation. And the reason that you do this is that any one of these is a potential copy of your clinical trial. And so it's important to run it many, many times, and then to look at the percentage of times that you get the endpoint that you're looking for, that you get the p-value or you get the response rate that you're looking for. That will give you an idea of the power of your trial to be able to be successful. I think this is a wonderful slide that I have used many, many times. It was created by my former colleague at Novartis, Dean Bottino, who's currently at Takeda Oncology. And to me, this really helps to understand how simulation works and how we can interpret simulation results. I happen to be from Miami, Florida, and especially in the latest weather, I think we're all very used to seeing hurricane maps that look like this. And what you see is these three little black dots represent the current path of the hurricane. And the blue line represents the most likely path for where it's going to hit my home state of Florida. If you look at the red, that is these are the bands of uncertainty, sorry. The red, yellow, and green are bands of uncertainty. So if you're in the red, you want to evacuate. If you're in the yellow, perhaps you want to board up windows. If you're in the green, you want to check again in 12 hours. And if you're outside of that, you should probably stay put. But what this is giving you is a band of uncertainty. You have the most likely course of the hurricane, that's the blue line, but you also have bands of uncertainty in which you want to make different decisions based upon where you are in that band. Very similarly, you do the same thing with drug and disease models. And this happens to be disease as a function of time, but you can see this in pretty much any kind of PK or PK/PD model, where the two black points represent our observed data. The blue line is the model through it and the most expected trajectory of that model. And then the red, yellow, and green represent decision points for us. So if we're interested in knowing whether or not we should stay on the current dose, whether we should monitor more closely, whether we should increase the dose or we should change therapies. And all of those decisions can be made by looking at these bands of uncertainty and deciding where is your patient and how sure do we need to be. So I really, really like this because to me it really hits home on the utility of simulations and helping us to make informed decisions. Speaking of uncertainty, I want to address some of the challenges that we face as modelers. There's a natural fear or distrust of things people don't understand, and particularly for modeling and other quantitative approaches, we often face some skepticism. Hopefully the nice slide from Dean that I just presented helps to put things in perspective, but I'd like to address it in another way that also reflects modeling and simulation as an integrative discipline. There is a universe of all possible answers out there to a question, but by integrating data, knowledge, from various sources into a model, you start to narrow that down. And at some point you might say this isn't narrow enough. You might be able to use the model itself to also help identify the gaps in information. Then you can seek out more data or design further studies to collect it. And those, once you add them, you narrow things further. Now, modeling may not have dropped that oval to a point, but it certainly facilitates a much smaller set from which to determine your outcomes. And so if a team and regulators can understand that, they should be more comfortable with some of the data-driven decisions that are made with modeling and simulation because we've drastically reduced the uncertainty. And in addition, a utility that we often forget is that a model can serve as a repository. It integrates all of the relevant data and information into a tidy package, and this can be used and reused for future data-driven decisions. So for us within the modeling and simulation discipline, what are the properties of a useful model? Well, first and foremost, it has to be able to characterize the observed data. So this is the descriptive component, and include the most important features of the data. In situations where, for example, we're interested in determining the exposure as a function of AUC, then getting the line through all the points, including the one at Cmax, may not be as critical as making sure that we're capturing the overall trend and the overall exposure to the data. Obviously, it needs to be logically consistent and biologically plausible. You don't want to end up making decisions that aren't clinically relevant or don't make sense in the context of use of that particular drug. It must be validated by empirical observations. When we talk about model validation, which I didn't get into in this presentation, a lot of times what will happen is we will develop the model on part of the data set and then validate it on another part to make sure that it's consistent with external data as well. That's called external validation. It has to have the appropriate level of precision and detail, not more. It is possible to model forever and make things much more perfect, but at some point you have diminishing returns. So you have to have the appropriate level of precision and detail, but not more. I mentioned in the previous slide that it serves as a platform for data and knowledge integration, and I believe that it's very effective as a communication tool because it helps to collapse a lot of information into a succinct form that can help us to describe in a graphical or in a single equation a lot of data. It may allow for extrapolation outside the data range based on certain assumptions, and I've talked in this presentation about both interpolation and extrapolation. And of course, when you work in pharma, it has to be completed on time and used by the study team. Now you'll notice that in the title it says properties of a useful model, and I put a star there. You may hear modelers use this quote, all models are wrong, some are useful. There's a lot of baggage behind this particular quote, and the problem is when people hear it and they don't fully understand what that means, what they hear is all models are wrong. So my colleague Pete has suggested a corollary that actually captures the same information but is maybe a little bit more palatable to modelers, which is all models are approximations. Some perform better than others. I like that actually better because no model is going to represent perfect reality. Even QSP models and their granularity don't necessarily represent every single part of the platform, of the pathway. So I think we need to be clear that models are approximations, but that they have a lot of utility if you use them responsibly. And of course, what modeling can't do is provide one true answer. As I showed you before with the narrowed possibilities, it's going to certainly drastically reduce your possibilities, but it may not give you the answer. It's going to get you closer though, and it won't explain everything. That level of granularity is important, but you may not be able to capture every component of your physiology or your pharmacology. It's not going to find an effect where there isn't one, and it's not going to give you the answers that you want. This has always been something that, as a modeler, I'm very, very careful not to overpromise. It's not going to make good studies unnecessary. It's not going to make your decisions for you. All of these things encapsulate the fact that we need to have data to model. We need to have scientific acumen. We need to have an understanding of the mechanism. We need to have support for our models. A model is not going to answer all of the questions for you, and certainly you need to have good studies. And similarly, it's not going to make a silk purse out of a sow's ear. A lot of people feel like garbage in, if you put it into a model, that suddenly you're going to have this beautiful truth. But unfortunately, garbage in, garbage out. So we want to ensure that we are properly using our data, and when we are coming up with our results and we're speaking to our teams and we're speaking to regulators, that we put the appropriate guardrails around the interpretation of that data so that it can be interpreted appropriately, and that we're not taking liberties with making assumptions that are outside of the data that we've collected. So in summary, pharmacometrics is a methodology that facilitates the succinct capture of data, knowledge, and assumptions over the lifespan of the project and across programs. So one of the beauties of modeling and simulation is that some models can be used and reused, particularly QSP and PBPK models can provide a platform for understanding both drug discovery and development. It allows us to quantify the properties of our compounds and predict new scenarios. Its execution requires skills in biology and clin pharm, mathematics, computation, and biostatistics. You do have a lot of modelers that come into the field more from the biostatistics or computational science, but they have to pair with somebody who has the biological and pharmacological understanding, because we don't want a model where the line goes through the points. The model has to represent reality to a certain extent. And so we need to make sure that we've got underlying physiological, pharmacological principles behind our models. Identifying and prioritizing modeling opportunities requires a combination of skills and dialogue. This is not a discipline where we sit in a room by ourselves and crunch numbers. That's part of our job. But a lot of our job is actually not the modeling itself, but the scoping, the understanding of the quantitative questions that need to be answered, and the interpretation of our modeling results. And that requires discussion, that requires education, and that requires collaboration with our team members. Successful implementation of pharmacometrics requires planning, dialogue between teams and modelers and between modelers. So there are sometimes technical issues that are difficult to solve, and we have a wonderful modeling community out there that can help us solve these problems. And of course, integration into the development process. I'm not going to go through this slide, but I've prepared some resources for you. Most of these has come from the International Society of Pharmacometrics. They have a page that has a listing of textbooks and literature, but I strongly recommend, if you're interested, to please check out ISOP as well as ACOP, which is our annual conference. And with that, I want to thank all of you for your attention and for your time. I want to thank my collaborators and those folks who have provided me feedback, given me slides, and given me some input on this presentation. I hope that this has been useful for you. I hope to hear from you if you have any questions or comments, but I hope that this has been a very good experience for you to learn about MIDD and that you enjoy the other modules going forward. Thank you so much for your attention, and I hope to hear from you.